Last recordings.
Okay, good evening everyone once again. Am I audible? Hello, all of you? Okay, fine, sir. Thank you. So, good evening once again. I am very much glad to introduce one more resource person. He is a scientist as well as a good teacher, voracious speaker by name called Dr. S. Umesh. Presently, he is serving as professor in biotechnology, University of Mysore. And he has earned his MPhil, MSc, PhD from University of Mysore only. And he has been elected to so many prestigious academies. Just to quote a few, Fellow of prestigious National Academy of Biological Sciences, International Society of Biotechnology, Indian Society of Mycology and Plant Pathology. And other than this, he has underwent a training, postdoctoral training in Denmark in 2002 under a prestigious program, DANIDA, at Virginia Tech University, Virginia, USA. He has also been invited to present his research findings in so many international and national conferences like Germany, Malaysia, and China. So, sir is serving as a professor, and he is taking subjects like microbiology, food and environmental biotechnology, and plant biotechnology. So, he is specialized in these fields. His research interests include plant biotechnology, molecular diagnostics of plant, animal, and foodborne pathogens. Also, it includes host pathogen interactions, cell and tissue culture technology. He is the coordinator of DBT-sponsored MSc Biotechnology program from our university. He has received so much of funds from various national and international funding agencies such as DST, DBT, UGC, ICMR, and East West International Seeds Private Limited, India. He has guided more than 25 MPhil students and 12 PhD students already been awarded PhD, and presently more than 8 students are working. And sir is also spent a lot of time to adjudicate more than 50 PhD theses around India. He has published 130 research papers, and his H index is 28, citation index, I-10 index is 50, and he has got more than 2,500 citations. He has also published one book on plant biotechnology published by TFRI New Delhi, and 5 book chapters. And he has been awarded Professor K.S. Jagdish Chandra Young Scientist Award from University of Mysore. With all these credentials, Professor Umesh work has been featured in Stanford University's global top 2% scientists based on the data obtained from Scopus. It is one of the important journal published by elsewhere in 2021. With this brief introduction, I welcome Professor Umesh sir to this session. Thank you, sir. Over to Dr. Umesh sir.
Thank you very much, Professor Basarajappa sir. Thank you. Thank you very much, Professor Basarajappa sir, for your nice introduction. Good evening, all of you. Am I audible?
Yes, sir. Good evening,
Yes,
sir.
sir.
Yes, sir.
Good evening, sir.
Okay, thank you.
Good evening, sir.
Okay. So, in another 1 hour or so, we will be discussing on innovative molecular diagnostics. That is my favorite aspect, mostly for detection of bacterial pathogens. Bacterial pathogens includes plants, animals, and human beings also. The diagnostics of any of the disease plays a major role in proposing the management of the diseases. because of this background information. The background information which I would like to tell you is, according to United Nations Organization, Food and Agriculture Organization, overall 30 to 35% yield reduction, or 30 to 35% of the total agricultural produce is going as waste because of pests and diseases. This is the statistics from United Nations Organization, FAO. They says, by any chance, if you can save so much of amount of loss, you can feed millions and millions of hungry mouths. And at the same time, FAO also says, on the global average, only 2 out of every 5 people are having the balanced 2 meals per day. That means remaining 3 people going to bed either hungrily or malnourished. Although we may surplus in producing the agricultural produce, the buying capacity is not there as far as Indian scenario is considered. On the other side, the disease resistance is a rule in nature, whereas susceptibility is just an accidental phenomenon. Arable land or agriculture land is shrinking day by day because of urbanization, industrialization, and other various anthropogenic activities. On one side, the total agricultural produce, about 30 to 35% is going as waste. On the other side, arable land is also shrinking on a global basis. That means technologists have a greater responsibility to produce the more and more food grains within the available limited agricultural land. And this is an also attempt to reduce the total loss of agricultural produce because of pests and diseases. Because if you want to propose any of the management strategy or control strategy, the pivotal importance is you have to diagnose, identify the disease causing organisms. If the diagnosis is going wrong, whatever secondary investments which we are proposing for management of the disease is going as waste. That is the reason, or hence, the proper identification of the disease is really important. With that background, our laboratory is involved in proposing major nucleic acid based diagnostics. The recent tendency is use of nucleic acid based diagnostics. Although the traditional diagnostics is you need to culture the microbe, the disease causing organism, or you have to raise the plant, look for symptoms, you have to culture the microbe, isolate the microbe from the diseased organism. That means a sort of proving Koch postulates. Robert Koch postulates there should be some organism to cause the disease, either in case of plants or animals or human beings. And that particular disease causing organism should be isolated and purified in the laboratory condition. And once again, the purest form of that isolated organism, which is responsible for producing the symptoms of the disease, should be reinoculated to the host plant or susceptible host plant. Then it should produce, we should be in a position to get the proper symptoms of the similar type which we observed earlier. This is the traditional or conventional identification methods, which are cumbersome, laborious, time consuming, involves a lot of land and other resources. And nowadays, use of nucleic acid based detection techniques is becoming a fascinating trend. Yes, sir. Anandan.
Are you moving the slides, sir?
No, not yet. Not yet. I'm just giving
Okay.
the introduction.
Okay. Okay, sir. Thank you. Sorry.
Okay. One of the important strategy to diagnose the disease is you need to properly identify the disease causing organism. If you want to propose the control strategy, it is a pivotal importance to properly diagnose the disease causing organism. If you properly diagnose the disease causing organism, it is easy to propose the control strategies or management strategies. Although there are various technology to properly diagnose the disease, many drawbacks or demerits of those technologies are there. That's why our laboratory is involved in use of DNA or genetic material for the proper diagnostics, for the proper identification of disease causing organism. That is how the innovative molecular diagnostics for the disease causing organism. And India is an agricultural country, and it is also called as fruit and vegetable basket of the world. because about 70% of the India's territory is occupied by agriculture. This is the basic statistics. And although there are many groups of pathogenic microorganisms like fungi, bacteria, and viruses, which are the major disease causing factors in case of plants, animals, and human beings, the fungal groups of microorganisms are responsible for reducing 15 to 20% of the yield loss, whereas bacterial group of microorganisms are responsible for 10 to 12% of the yield loss in the agricultural produce, and viral groups of microorganisms are responsible for 8 to 9% of the yield reduction. And in our laboratory, we mainly concentrate on only bacterial pathogenic detection. Bacteria perhaps next to fungi, rank next to fungi when damage of the crop plants is considered. And bacterial diseases can be devastating since bacteria possesses enormous amount of multiplication rate. Although bacteria cannot pierce the plant cell wall and get entry into the system, they always look for the natural openings like stomata or hydatodes or nectarithodes to get entry into the system. And once if it is entered, immediately in the intercellular spaces, they multiply enormous amount, and since the number of bacterial cells will be enormous, they move all parts of the plant body. While doing so, they may clog the xylem and the phloem, the main roads of the plant body, because of the exopolysaccharides present on the bacterial cell wall. And once if they clog, obviously the other part of the plant body is suffering from either water or food, so that the wilting takes place. And lack of effective control measures makes the disease management cumbersome and economically not feasible. Since we have also used some of the food material for the identification of foodborne pathogens. In this study, this is the 10 people's PhD work which I am presenting. Some of them have standardized the protocols for fungal, the plant pathogenic bacteria. Some of them have used foodborne pathogenic bacteria, and some of them have used domestic animal pathogenic bacteria. Food is also a complex substance derived from plant and animal source, which is consumed for nutritional support of an organism. We have used the food for isolation and characterization of foodborne pathogenic bacteria. Food contaminated with a number of pathogens transmitted by various routes and cause major health issues, which leads to life-threatening diseases, human diseases. Number of foodborne pathogens are reported by World Health Organization and CDC, Committee on Disease Control. Among them are Campylobacter, Listeria, Salmonella, Shigella, Staphylococcus aureus, Staph aureus have been recorded as the most frequent cause of food poisoning. This Staph aureus colonized approximately, because we have used the proper diagnostics for Staphylococcus aureus detection in case of foods, both vegetarian and non-vegetarian foods. Staph aureus colonized approximately 30% of the human population and resides predominantly in the anterior layers, harmlessly on the skin, and in GI tract, gastrointestinal tract. And needless to say, more than 90% of the microorganisms are beneficial to human beings, and only less than 10% of the microbes are harmful, which are responsible for causing diseases in case of either human beings or plants or domestic animals. And this Staphylococcus aureus causes skin and soft tissue infections, aggressively into bacteremia and infective endocarditis or sepsis. Identification through conventional methods are replaced by nucleic acid tests, NATs we call, due to lack of sensitivity, laborious and complex to perform. Identification of pathogens in a short period of time is still challenging to the scientific world, and developing easy and sensitive method for identification of pathogens can give more attention about food safety, and therefore they are released for the consumption. All these data which were published in the critical reviews of food science and nutrition, and also Indian phytopathology, mostly pathogenic plant pathogenic bacterial review. Koch postulates is one of the basic important diagnostics. As I said, we need to look for symptoms in case of plants or animals or human beings, and we need to isolate the disease incitant or disease causing organism, and we need to purify. You need to grow the microbe other than the host under artificial laboratory conditions. Then we need to purify, and the pure form of that particular pathogen should be re-inoculated. These are the postulates. And importance of diagnostics, because I told you about 30 to 35% of the total agricultural produce is going as waste because of pests and diseases. And today, diagnosticians have an array of methodologies, either traditional or conventional or nucleic acid based or immunological techniques. Different kinds of diagnostic techniques are there. Traditionally, cultural methods are mostly being practiced, which are none other than the cultural methods. Mostly you have to culture those disease causing organisms which are relatively slow process. Nucleic acid based amplification systems like PCR are the promising methods for rapid identification of low copy numbers, low numbers of plant pathogenic bacteria. The number of disease causing organism cell even at the lowest concentration which can be able to detect. But the main drawback of this particular PCR is even the disease causing organism is not viable. Even if it is dead, it shows the positive reaction. That is the only drawback of using the PCR, because it will not differentiate the pathogen whether it is viable or dead. So what are the host pathosystems which we have used in the current study? We have used Xanthomonas xenopodis respiratoria because this is bacterial spot causing pathogen in case of chili, tomato, and the brinjal. Xanthomonas oryzae, bacterial leaf blight causing disease in case of rice, bacterial leaf blight disease. Xanthomonas campestris in cabbage, block rot of cabbage, and one of the most important deadly pathogenic bacteria is Ralstonia solanacearum, which will cause the wilt disease in 450 plants of more than 50 botanical families. This has got very, very wide host range. Ralstonia solanacearum has got very wide host range, which is bacterial wilt causing pathogen in case of chili, tomato, brinjal, potato, tobacco, you name any plant, Ralstonia solanacearum will be causing the disease. That's why it is called as it has got very wide host range, which can cause the disease in among 450 different plants, which are spread across more than 50 botanical families. And we have also used foodborne pathogen, that is Staphylococcus aureus, and the domestic animals, the zoonotic disease, Leptospira, and the Brucella, which we have used. Both these diseases are zoonotic diseases, which will cause the disease in case of domestic cattle, and the people or the human beings which are coming across with this zoonotic infected cattle, that disease will spread. That's why the disease spread from the animal to human being and vice versa is zoonosis. These are the worst pathosystems which we have used in our present investigations. These are the typical symptoms of tomato plant, showing typical symptom of bacterial spot disease caused by Xanthomonas xenopodis, and tomato plant showing typical symptoms of bacterial wilt disease caused by Ralstonia solanacearum, and tomato fruit showing typical symptoms of bacterial spot disease. And Xanthomonas campestris, which is mostly, these are the V-shaped lesions on the cabbage plant. Initially, why these V-shaped lesions? Because during the early morning hours, there is water droplets. The guttation pores will be there through which water exchange takes place. And on that particular spot, if by any chance bacteria come across, immediately it will enter into the host system and multiply by causing these lesions. This is the entire field which is showing block rot disease in case of cabbage. Similarly, bacterial leaf blight disease in case of rice plants. The distal leaf, the distal end of the leaf becoming yellowing, and it will progress towards the basal region of the plant. This is none other than Xanthomonas oryzae, which is bacterial leaf blight causing disease, which has got the yield loss up to 70%. Report says 25 to 70% of the yield reduces of rice because of this particular disease. And these are the degree of infection from the distal part of the leaf towards the basal part of the leaf. This is leaf clipping method, which is being standardized by International Rice Research Institute, Philippines, Manila. And this leaf clipping method, it can also be used as a parameter to assess the degree of susceptibility and the degree of resistance among different rice cultivars. This is highly susceptible variety, where within 24 to 48 hours, the entire leaf has become yellow. These are moderately resistant or moderately susceptible rice cultivars. So this leaf clipping is a method to assess screening of host differentials for their degree of resistance or degree of susceptibility as far as bacterial leaf blight disease is considered. This bacterial leaf blight disease is caused by Xanthomonas vorizae, Pathover vorizae. So traditionally, isolation and characterization of different plant pathogens using biochemical and the molecular methods. We have procured the seeds from different sources and we have screened those seeds and planting material using the standard procedures of International Seed Testing Association, headquarters at Zurich, Switzerland. And we have characterized the isolated pathogen by subjecting morphological characterization test, biochemical or physiological tests like Gram reaction, KOH solubility, gelatin hydrolysis, starch hydrolysis, oxidase reaction, all these things. And in addition to that, we have also subjected the isolated pathogen for molecular detection using 16S rRNA primers and also species-specific primers using the PCR amplification techniques. These are the morphological characterization of different bacterial pathogens. Colony morphology of Xanthomonas campestris, Pathover campestris, which is a block rot causing pathogen in case of cruciferae, whole vegetables, and colony morphology of Xanthomonas hexanopodis, Pathover vesicatoria, which is bacterial spot causing pathogen in case of tomato, chili, and the brinjal, and colony morphology of Ralstonia solanacearum. This colony, this bacteria is highly considered as highly deadly disease causing organism because this can cause the disease in about 450 plants which spread across 50 botanical families, and this is Xanthomonas vorizae, Pathover vorizae, which is bacterial leaf blight causing pathogen in case of rice. And now exactly the polymerase chain reaction starts. We have slightly modified this polymerase chain reaction using different techniques like single strand conformation polymorphism PCR. That is, PCR SSCP is mostly used, widely used technique for identification of point mutations in case of various organisms due to their simplicity, sensitivity, and rapidity. The 16S rRNA gene whose use is considered as the gold standard for bacterial identification. Single strand conformation polymorphism analysis of PCR amplified products of 16S rRNA gene has more recently been used as an alternative genomic sequencing, alternative to genomic sequencing for the identification of different pathogenic bacteria. The procedural details of PCR SSCP is very simple. After the amplification of PCR products, the target sequence, the amplified product is denatured to 2 single-stranded DNA, SSCP, single-strand conformation polymorphism, and further subjected to non-denaturing polyacrylate gel electrophoresis. Under non-denaturing conditions, single-stranded DNA as a secondary structures. These are the secondary structures which are specific folds. These specific folds will be unique to subspecies level, not only genus and the species level. The secondary structures is unique to subspecies level. Under non-denaturing conditions, single-stranded DNA as a secondary structure that is determined by the nucleotide sequence. The mobility of the single-stranded DNA depends on the secondary structure of the amplified product. So, PCR single-stranded conformation polymorphism technique, the procedural details, we have to isolate the DNA from bacteria and we have to amplify that particular DNA from bacterial pathogenic organisms using 16S rRNA primers, and we will get PCR SSCP fingerprints, and further PCR SSCP fingerprints will be compared with other phytobacterial pathogens for validation. This is the amplification of DNA, and this is exactly the PCR SSCP fingerprints of Ralstonia solanacearum. We have used many different isolates of Ralstonia solanacearum. All those Ralstonia solanacearum isolates, which gives a unique bending pattern. This is the fingerprint, PCR SSCP fingerprint of one particular pathogen. PCR SSCP fingerprints of Xanthomonas hexanopodis, Pethorvis catoria. And this is PCR SSP fingerprints of Xanthomonas oryzae, Pethorhizae. And when we compare different pathogenic organisms, this is the banding pattern. This banding pattern is unique to this particular Rhamnosus solanacearum. This will be unique to Xanthomonas Xenopodis Pethorvis catoria, Xanthomonas oryzae, Pethorhizae, and the positive and the negative controls. The application of this PCR SSCP, because SSCP has simplicity in usage without the requirement of any of these special techniques, because only PCR machine we need to use. And though the DNA sequencing has become the common technique nowadays, the equipment is still requires not in common laboratories. And this entire data we have published in Acta Biophysica, Biochemica Sinica in one of the important journal. And after this SSCP technique, we have also used colony PCR SSCP. In this normal PCR SSCP, we need to isolate the DNA. But here in the colony PCR, DNA need not have to be isolated. Directly, we can use the pure colony of bacterial pathogens. And since we are using 16S RNA primers, and also for the confirmation, we are using the species-specific primer. If the particular pathogen is present in the culture, the pure culture, it will amplify, otherwise it will not amplify. So colony PCR requires isolation of pathogen, but not isolation of DNA. And therefore it is rapid, accurate, and cost-effective approach for identification of the particular pathogen. Colony PCR SSCP has an advantage because of its rapidity and simplicity. The procedural details, we need to get the culture of bacteria. We need to amplify with the help of 16S RNA primers. You need not have to isolate the DNA. And we need to subject the SSCP fingerprinting pattern. And those fingerprints will be compared for the ready reckoner, the standard DNA fingerprinting technique. And this is colony PCR SSCP fingerprints of different pathogenic bacteria we have used, particularly in case of Rhamnosus solanacearum isolates. Without isolating the DNA, directly we can use the pure culture. Colony PCR SSCP of Xanthomonas Xenopodis, Pethorvis catoria, and colony PCR SSCP banding pattern of Rhamnosus solanacearum when compared to Xanthomonas Xenopodis, Pethorvis catoria. So these are the techniques, the modification of PCR for the proper identification of the pathogenic bacteria. The application of colony PCR SSCP, which has been developed, is a qualitative tool for the detection of pathogen that can overcome the isolation of DNA. Direct colony PCR SSCP for the detection of Xanthomonas Xenopodis, Pethorvis catoria, and Rhamnosus solanacearum is relatively rapid when compared to the conventional isolation techniques and morphological examination of isolated bacteria. The other modification of PCR is multiplex, because we have used single strand conformation polymorphism, the first one. The second one is without isolating the DNA, directly you can use the mixed culture, that is colony PCR. And the other modification is in the same PCR reaction, is it possible for us to detect 2 or 3 or 4 pathogenic bacteria? That is in the form of multiplexing of pathogenic bacteria, multiplex PCR. Multiplex PCR is a widespread molecular biology technique for the amplification of multiple targets in a single PCR experiment, so that you can reduce the usage of reagents. More than one target sequence can be amplified using multiple primer pairs in a reaction mixture. Our present research exploits this method for identification of pathogenic bacteria like Xanthomonas campestris, Pethora campestris, which causes block rot of cabbage, and Rhamnosus solanacearum, which causes bacterial wilt of tomato. So here in case of XCC and cabbage, we have attempted to identify both host and the pathogen in the same PCR reaction. Similarly, Rhamnosus solanacearum along with its host. These HRP genes we have used for multiplexing, hypersensitive response and pathogenicity genes, HRP genes, which direct their ability to elicit hypersensitive response. And these HRP genes encode a type 3 protein secretion system that appears to be capable of delivering AVR proteins, avirulence proteins, across the walls and the plasma membranes of living plant cells. The multiplex PCR fingerprints were obtained which amplified a 619 base pair fragment of HRPF gene from XCC pathogen and a 360 base pair section of ITS region from Brassica species. This is the point which I am referring to. We have identified both host and the pathogen in a same PCR reaction. Pathogen amplified at this particular base pair, whereas host amplified at this particular base pair. So this is a sort of multiplexing identification of both host and the pathogen in the same PCR reaction. This multiplexing helps to identify the mixture of pathogenic bacteria and along with their respective host material. This will be applicable more important in case of foodborne pathogens. The procedural details of multiplexing PCR is very simple. We need to isolate the disease causing organism, and for traditional way we need to confirm by subjecting it for morphological biochemical characterization test, and we need to confirm that particular pathogen by amplifying the species specific primers. Block rot pathogen amplified a typical 619 base pairs typical to Xanthomonas campestris, Pathover campestris in comparison with other xanthomonads. This is the amplified genetic material, and multiplex PCR reaction we need to isolate the plant pathogen and the biochemical characterization. We need to confirm the pathogen by subjecting it for various morphological and biochemical characterization techniques, and at the end, after confirmation, we have used this multiplex PCR for both Ralston solanaceae amplification and also Xanthomonas oryzae, Pathover oryzae amplification, which are two different pathogenic bacteria which causes a disease in case of two different host species. Excuse me. This is the PCR amplification of Xanthomonas oryzae, Pathover oryzae with a species specific primers when compared to the molecular weight marker, and PCR amplification primers where Ralston solanaceae. When we amplify the Xanthomonas oryzae pathogen at this particular wavelength base pairs, and when we amplify Ralston solanaceae at this particular base pair. When we combine these two with the help of multiplexing, this is the multiplex PCR profile of Ralston solanaceae and also Xanthomonas oryzae. Here, in case of multiplexing, one of the important criteria is adjusting melting temperature. TM adjustment is very, very difficult. We need to subject for, we need to give various trial and error methods to arrive at one particular melting temperature. For example, Xanthomonas oryzae at 61 plus minus 1 degree centigrade and Ralston solanaceae at 57 plus minus 1 degree centigrade. In between this 57 and 61, we have used for the amplification at this particular temperature of 58 and 64. So adjusting the melting temperature is very important in case of multiplex, because that annealing temperature should be exactly in correlation with the particular amplification of individual pathogenic organisms. It should not hamper. The application of this multiplex PCR plays a crucial role in the identification of several pathogenic organisms in a single reaction, so that we can reduce the usage of the reagents, expensive reagents. The procedure developed from the present studies overcomes the laborious traditional or conventional characterization of phytobacterial pathogenic organisms. This data we have published in this particular journal. And the, excuse me. The application part of this multiplex, seed samples can also be directly subjected to multiplex, because if the pathogen is there in the seed sample, it will amplify. If it is not there, it will not amplify. This can be used to identify properly the plants along with the pathogens and also the mixture of pathogens. If we use the amplification of 2 targets, it is duplex PCR. The amplification of 3 targets, it is triplex PCR. And the amplification of more than 3 targets, either it could be from different pathogens or it could be from different hosts, then it is multiplex PCR. Amplification of different targets, 3 or more targets in a same PCR reaction. This we have published in all these journals. So development of diagnostics for leptospiral identification. Leptospira, which is a zoonotic disease, which causes the disease in case of human beings and also in case of domestic cattle. So we have isolated the DNA from the leptospiral individual species of Leptospira. We have designed in our laboratory the primers which are targeted for amplification of this Leptospira, and we have subjected this amplification through PCR, and we have characterized the amplicons, transformation, plasmid isolation, and ultimately we have subjected the polymerase chain reaction single strand conformation polymorphism for confirmation of leptospiral species. So these are the primer pairs which we have designed in our laboratory for amplification of Leptospira species. This LIP-L32, LIP-L41, LIP-L is lipoprotein L, which is the coat protein which can be used to amplify the coat protein of this Leptospira bacteria along with the 16S rRNA primers. These are the standardized thermal cycling conditions for the amplification of Leptospira. As I said, it is a zoonotic disease, and this is re-digestion of Leptospira LIP-L32 and LIP-L41 genes. These are the PCR products of Leptospira isolates amplified with lipoprotein L32 primer and lipoprotein L32 gene of Leptospira re-digestion. And the PCR products of Leptospira isolates amplified with both the genes at this particular base pairs 427 and 263, all these things. And re-digestion, restriction digestion, preparation of ligation mixture, followed by the procedural details of competent colonies, and insert release of lipoprotein L31, similarly lipoprotein L32 gene, and ultimately we have subjected the amplified product for SSCP analysis. PCR products were first evaluated for their purity by agrosteel electrophoresis, and we have used 5 microliter of individual PCR products were mixed with 25 microliter of denaturing buffer. The mixtures were heated. This is the heat shock treatment at 96 degrees centigrade for 10 minutes, then chilled with ice cold, because we need to provide both heat and cold shock to anneal double-stranded DNA into a single-stranded DNA. Denatured PCR products were loaded onto non-denatured gel. This is the banding pattern, the fingerprinting of Leptospira isolates amplified with 16S rRNA primer. And these are the different Leptospira species: Leptospira hictioemerae, Leptospira javanica, and different Leptospira species, along with the positive and the negative controls. So PCR SSCP banding patterns of Leptospira species. Banding pattern was visualized on non-denaturing polyacrylamide gel. Isolates 1 to 8 are different species of Leptospira. Leptospira hictioemerae, Leptospira javanica, and things like that, along with the positive and the negative control, along with molecular weight marker. And this data we have published in this particular veterinary science and technology journal, since it is related to veterinary disease. And foodborne pathogens, particularly isolation and characterization of Staphylococcus aureus, Staph aureus from food samples. We have collected various food samples: vegetarian and non-vegetarian, dairy, bakery products, processed foods. These are the different food samples from the vegetarian section, different dairy samples, different dairy food samples, bakery items, processed foods, and non-vegetarian items. These are the number of isolates and representative isolates used in the present study. Use of MALDI biotyper is an important tool. This is first of its kind, because only MALDI biotyper used to identify human pathogens. We have used MALDI biotyper for the isolation and characterization of foodborne pathogens. The procedural, the entire details are collection of different food samples, isolation of Staphylococcus aureus, and one part of the isolated Staphylococcus aureus is subjected to MALDI biotyper, and the other part is biochemical characterization, and subsequently the molecular approaches, use of DNA extraction followed by amplification of PCR with the specific primer and also with the universal primer. And we have used the SSCP analysis for confirmation of Staphylococcus aureus, and most importantly, matrix assisted laser desorption ionization biotyper which we have used in two different ways. One is direct smear and the other one is extraction of protein. And subsequently, we need to use that equipment for identification of Staphylococcus aureus. So these are the different food samples which are associated with the foodborne pathogens from vegetarian, non-vegetarian, dairy products and the bakery products, Staphylococcus aureus, Bacillus, E. coli, Vibrio, things like that. And these are the colony morphology or morphological characterization of foodborne Staphylococcus aureus and Shigella flexneri, Salmonella typhimurium, Bacillus aureus, E. coli, and the other foodborne pathogenic microorganisms. How exactly they look like on the specific chromogenic medium, on their selective or the semi-selective medium. Selective or semi-selective medium is the careful combination of the required nutrients for particular growth of one particular pathogen, which will selectively supports the growth of one kind of organism, and it will selectively suppress the growth of other organisms. The staphylococcal species associated with the different food items. These are the vegetarian, non-vegetarian food items and foodborne staphylococcal species on the selective medium and their biochemical characterization of foodborne Staphylococcus aureus like Gram reaction, catalase test, Kovacs oxidase, KOH solubility, starch hydrolysis, gelatin hydrolysis, and the other tests. Followed by these are the various isolates of Staphylococcus aureus and their we have isolated the gene and amplified with the 16S RNA primers, and we have also subjected for the sequence submission to gene bank and obtained the accession numbers from the public domain. So moldy biotyper in the identification of pathogen. How exactly this instrument, either you can use the sample culture smear or the mixture on the moldy tough MS plate, and the moldy instrument we need to feed this plate and it will give us the spectra, and this spectra which is comparing the library which is already there in the instrument and give us 99% homology to one particular organism, or 98% or 60% or 70% or something like that. By giving this range, if the reading is 2.3 to 3, this is highly probable species identification along with the color and the symbols. All these three parameters can be used for the probable identification of the particular species. And the reading range from 2 to 2.29 secure genus identification and species is probable identification with this particular 2+ and the green color. And the other readings are like this. So the identified scores values meaning for analyzed samples in the moldy biotyper, matrix assisted laser desorption ionization biotyper. These are the multi-tough MS profile of representative protein spectra of Staphylococcus aureus, different Staphylococcus aureus species which we have used for identification. These are the different foodborne pathogenic organisms. When we subject them for moldy biotyper analysis like E. coli, Bacillus cereus, Vibrio, Drexler, etc. And when we use the staphylococcal strains discriminated by universally conserved NUC thermonuclease gene, and the specific identification of Staphylococcus aureus was assessed by species-specific primers, because once if we identify based on moldy biotyper, subsequently we have confirmed those identified organisms by using other conventional technique also, and by amplification of species-specific genes in our routine experimental procedures, and also PCR SSCP analysis. And these are PCR SSCP fingerprints of different foodborne pathogens. So these are different foodborne pathogens, Staphylococcus aureus, Bacillus cereus, Vibrio, and all these representative isolated which are given the name as UOM 012, University of Mysore 01218, something like that, because we have subjected almost 500 different foodborne pathogenic microorganisms, particularly foodborne pathogenic bacteria for moldy biotyper, and we have given here the data of only representative isolates. So we have successfully characterized the foodborne 160 methicillin resistant Staphylococcus aureus, and in the present study involving 50 different food samples showed the representative moldy biotyper. Although we have submitted the gene bank accession more than 500 different samples, which we have given here only 50 or so. We have developed a PCR SSCP, single polymerase chain reaction, single strand conformation polymorphism for the first time to discriminate the foodborne pathogens at the species level in the present investigation. And this data we have published in the Nature scientific reports, and immediately this particular person got the postdoctoral fellowship in USA. And LAMP assay, loop mediated isothermal amplification. We have used one more, we have standardized in our laboratory the one more assay, loop mediated isothermal amplification, with the selection of SPA as the target gene. We have designed the primers for this identification of the foodborne pathogenic bacteria, and this LAMP reaction we have standardized, and we have tested the sensitivity of the LAMP, and subsequently we have analyzed, because the entire idea of standardizing procedures for use of nucleic acid based detection techniques, because traditionally we need to culture the organisms, or we need to cultivate the plants, raise the seedlings, look for typical symptoms on the seedlings, then isolate the pathogenic organism from the diseased seedlings or diseased plants, and then isolated pathogen should be purified in the laboratory and subjected for various morphological, biochemical and physiological tests, also used by immunological assays for identification of these disease causing organism. And in our study we have used the DNA based amplification technique, which is very effectively, which we can use more than thousands of different samples at a stretch. Within a day or two, you can assess whether this particular seed sample is free from any of the pathogenic organism, or the target pathogen is there in this particular seed or not. Like that, these tests are easy to perform, and which we can subject for a fast detection assay. So these are the primers which we have used for LAMP primers, loop mediated isothermal amplification, and the LAMP condition, schematic representation of loop mediated isothermal amplification procedures. So this LAMP assay, both in-gel assay and in-tube assay, we have standardized. In-gel assay also used to amplify a particular target gene from any of the disease causing organism. Although we have used only the representative pathogenic microorganism from the plants or from the food, which we can extrapolate for the organisms of any of the human pathogens or plant pathogens or animal pathogens, domestic animal pathogens, whether it is from fungi or bacteria or even viruses. We can apply the same techniques for identification of any of the disease causing organisms. Although here I am showing only bacteria as a representative pathogenic organisms. So this is the isolated Staphylococcus aureus isolates were confirmed by LAMP F3B3 primers. This LAMP assay is loop mediated isothermal amplification. So these are the reaction components, like 0.2 micromolar of the primers, that is forward and the reverse primers, 0.8 micromolars of loop primers, and the BST DNA polymerase large fragment, DNTPs, magnesium sulfate and other required reagents. These are the reaction components for loop mediated isothermal amplification. LAMP assay, as I said, LAMP assay can be done both in gel assay and the in tube assay. In gel, you can rise as usual polymerase chain reaction followed by page polyacrylamide gel electrophoresis, and the same reaction which you can visualize on the tubes, Eppendorf tubes. Eppendorf tubes can also be used so that this particular LAMP assay can be performed at a very faster rate. So entire idea of using DNA based nucleic acid detection techniques is to perform at a faster rate or within less amount of time. You can screen hundreds and thousands of pathogenic microorganisms, or it can be applied to seed industry also, because seed industries are producing the good quality seeds for export business and also for domestic use. For all those seeds, we need to subject all those seeds before they release into the commercial purpose, commercial market, whether these seeds are free from this pathogen or this pathogen or this pathogen. So export business, if you want to take up any seed company, these certificates issued by ISTA, International Seed Testing Association, or ISTA accredited labs also able to provide the certification, which will be helpful for taking up the import and the export business of seeds, because seeds is a multi-million-dollar industry now. Sometimes seeds are costlier than gold. Few grams of seeds of tomato, which is expensive than the gold. So these are the bacterial reactions, bacterial strains which we have used for LAMP assay, and specificity of the loop mediated isothermal amplification assay for the detection of Staphylococcus aureus. This particular A is in gel assay and the B is in tube assay. I was just referring, this LAMP assay can be conducted in gel as well as in tube level. So once if you have standardized this in gel assay, this in tube assay can be performed very easily. Once if you standardize the amplification product of this LAMP product, similarly this color change in the gel in the tube can give exactly similar to those of in gel assay. So the specificity of the LAMP assay for the detection of Staphylococcus aureus, this indicates the specificity of the LAMP assay detecting the Staphylococcus aureus in the artificially contaminated E. coli, because this particular 200 base pairs and 100 base pairs amplification, and the confirmation of the LAMP products by this HNB sign, blue color for the positive samples on the 2% agarose gel. So in tube assay can be also used for amplification reaction. So this loop mediated isothermal amplification is efficiently used to screen many different seed samples within the less amount of time. All these techniques which we have standardized are exclusively meant for screening the samples, hundreds of samples with a less amount of time. That is our target, because otherwise what is the procedure? You need to rise the plant. Whatever the seeds which you are subjected for export import business, you need to draw the sample using ISTA standardized procedures. International Seed Testing Association procedures we need to subject. We need to draw the sample, and those are primary samples. Then you need to pool all the primary samples to get the composite sample. From the composite sample, you have to submit to a particular laboratory as a submitted sample. And from the submitted sample, you have to draw as a working sample. And from this working sample, you need to rise the crop, or you need to plate the seeds on the moistened blotted disc. That is standard blotter technique we need to use for expression of any of the seed-borne pathogenic organisms. Either it is seed-borne bacteria or seed-borne fungi. For seed-borne fungi, standard blotter method is being used. For seed-borne bacteria, you need to plate the seeds on the selective medium. Plating the seeds on the selective medium or plating the seeds on the generalized medium. Either nutrient agar medium can be used, or sometimes species-specific selective medium can also be used for plating the seeds and look for the appearance of the bacterial colonies are surrounded by the seeds. And these bacterial colonies you need to isolate to the laboratory condition, because you may get mixture of different bacterial colonies, and you need to purify those bacterial colonies from a mixture, and individually you need to subject for those individual pure cultures for various biochemical, morphological, immunological or DNA based techniques. And sometimes an individual selective medium or semi-selective medium, which I told you, which will sequentially separately enhances the growth of a particular bacterium, and it will also suppress the growth of other bacterium which is not targeted. Most of the saprophytic bacterium will be reduced, will be suppressed by this semi-selective or selective medium. So when that is the procedure, then you need to isolate, confirm, and then inoculate to the host plants to get the similar symptoms. Entire thing will take minimum of 3 months, 3 to 6 months time. Whereas use of DNA-based nucleic acid detection techniques within 2 days or within 3 days, large number of seed samples can be screened. Of course, the drawback of this use of DNA-based technique, which I told you, even if the pathogen is dead on the seed sample, even if the pathogen is dead on the plants, even if the pathogen is dead on any of the source, it will give the positive reaction. So that is the only drawback of use of DNA-based nucleic acid detection techniques. All other techniques, which will very clearly gives us whether the pathogen is alive or dead, because the seedling symptom test, you need to raise the susceptible plants, inoculate the susceptible plants to the purified pathogenic organism to get the similar symptoms, seedling symptom test. And if you get similar symptoms, then you can ascertain the isolated pathogen is Xanthomonas or Pseudomonas or Clavibacter or something like that. But use of DNA amplification, even if the organism is dead, such organisms' DNA also amplified. This is the only drawback from the use of any of these DNA-based, nucleic acid-based detection techniques. So sensitivity of conventional PCR assay and in-tube LAMP reaction. LAMP is loop-mediated isothermal amplification reaction, and along with the 10-fold serial dilution of Staphylococcus aureus. Even if minute concentration of pathogenic propagule is there on the sample, which will give the reaction. So we have compared many different the concentration of these are the different concentrations which we have used. Up to this concentration level, the reaction is positive. So in gel assay and also in tube assay can be exactly compared for loop mediated isothermal amplification reactions. I told you the DNA based amplification test, the only drawback is even if the pathogen is dead, it will give the positive reaction. So we have developed a versatile loop mediated isothermal amplification test, that is the LAMP method, LAMP assay for identification of Staphylococcus aureus. This data we have published in the Medicinal and the Chemical Communications, Food Research International, because most of the items are related to food, and recently in the year 2020, Arabian Journal of Chemistry also published our data. So the overall strategy of our molecular diagnostics is a newer approach for identification of disease causing organisms. Although this is just an experimental basis which we have used pathogenic bacteria, plant pathogenic bacteria, and we have isolated the pathogen from the domestic animals, and also we have isolated the pathogen from food samples. The same detection techniques can be also used or can also be extrapolated for any pathogen, either it is from bacteria or from fungi or even from the viruses. Although we need to slightly alter the procedural or melting temperature in case of PCR or the primers in case of PCR. All these minor adjustments we need to do for identification and characterization of plant pathogenic, food pathogenic or domestic animal pathogenic bacterial organisms. So as I said, other than this nucleic acid based detection techniques, although they are the gold standards, but because of laborious nature, because of involvement of the land, the technical personnel or the preparation of land, because to sow the seeds, raise the crop, then look for the symptoms, then isolate the pathogen from the symptomatic portion of the plant body, then purify, then you have to re-inoculate to the susceptible host to ascertain or to confirm whatever isolated pathogen is either Ralstonea solanacearum or Clavibacter or Pseudomonas or Xanthomonas. Whereas here in case of nucleic acid based, which can overcome the laborious or the time consuming procedures, because you need to isolate the DNA sometimes and you need to amplify the DNA based on whatever primers which we are using to amplify. But even environmental samples, without knowing the identity of the pathogen, if by any chance, if the soil sample is suspected to be infected with Pseudomonas or suspected to be infected with Xanthomonas, directly you can use a minute concentration of soil sample for the amplification of species specific primers. If the pathogen is there in the soil sample, it will amplify. a sort of metagenomic approach. Metagenomic approach is nothing but identification of unknown organism, which is not possible to culture because just soil sample we have used or just plant debris we have used, but the primers which you have used should be the suspected pathogenic primers. By any chance, if you subject a particular soil sample is infected with, or if you suspect the particular leaf sample is infected with one particular bacterium, that particular bacterium's primer if you use for the amplification, then obviously you can confirm this soil is infected with this Xanthomonas or this leaf is infected with this Pseudomonas. A metagenomic approach is also important for isolation and confirmation of pathogenic bacterial organisms. So whatever standardized procedures in our laboratory, which we can use for isolation and confirmation of disease causing organisms in general. But since we have used only pathogenic bacterial organisms, all these pathogenic bacteria can be amplified with our known PCR primers. Although the 16S rRNA genes, amplification of 16S rRNA gene, which is considered as the gold standard for prokaryotic identification, because 16S rRNA gene is universally present in almost all prokaryotic organisms, which is sufficiently conserved from decades together, from centuries together, which is conserved and which is of adequate length. Because of these reasons, 16S rRNA gene is used as the gold standard for identification of prokaryotic organisms. On the contrary, 18S rRNA gene, which is used, the amplification of 18S rRNA gene, which is used as gold standard for the amplification of, for identification of eukaryotic organisms. So prokaryotic organisms, 16S rRNA, and the eukaryotic organisms, 18S rRNA. That is how we have used DNA amplification for the identification and the confirmation of prokaryotic organisms like bacteria. And also the same detection techniques can be used to identify unknown or non-culturable organisms which are present in the environment, either soil sample or plant debris or any of the environmental sources. So this is in brief the use of nucleic acid based detection techniques, a novel approach of DNA based detection techniques for identification and confirmation of disease causing organisms. As I said, this we have used only to amplify pathogenic bacteria. It can also apply to amplify either human pathogens or fungal pathogens or viral pathogens. But only thing is we need to change the melting temperature, the primers, and accordingly we need to standardize the PCR procedures. I'll stop at this stage. If you have any questions, you can feel free to ask me. Thank you.
Hello, sir.
Yes.
Hello.
Yes, sir.
Yeah, sir. In the slide, I have seen that you have mentioned that the LAMP has more sensitivity compared to PCR.
Yeah.
Yeah, because it can even detect at 10-fold dilation, whereas PCR fails to detect.
Yeah.
So, whether this is because of high specificity of PCR or any other reason is there?
No, it is because of high specificity of PCR. This is high specificity of PCR. Sometimes it will not amplify.
Okay, thank you.
Yeah. Thank you.
Hello, sir.
Yes, sir. Yes, madam.
Sir, I'm Kavya from Central University of Tamil Nadu.
Okay.
Sir, very nice talk, sir. Thank you for that.
Thank you, madam.
Sir, I have a question. In your LAMP assay, you have used 3 sets of primers, outer, inner, and loop primers.
Yeah.
I observed that in the PCR mix, you add them in different concentrations.
Yeah.
I was wondering what might be the reason.
No, we have standardized the different concentrations.
Okay.
For standardization protocol, for the standardization purpose, we have used different concentrations.
All right. Okay, sir. But if I'm going to apply this for some other organism, this may not be the case. I have to standardize again.
Exactly.
Okay.
Yes.
Thank you.
Yeah.
Hello, sir.
Yes, madam.
Thank you so much for the session, sir.
Yeah.
Sir, you talked about colony PCR, no, sir?
Colony PCR, yeah.
Yeah. Sir, those colonies need to be blotted, sir, during this technique.
Actually, sometimes mixture of different colonies can be used or purified colony of a particular organism. You need not have to isolate DNA. You
Okay,
can
sir.
avoid, you can skip the step of isolating DNA. Directly, you can use the colonies.
Yes, sir, that's what. These colonies to be blotted onto membrane or directly?
No. Directly, you can take the smear of colony with sterile distilled water dilution of 10 microliter or 20 microliter. Directly, you can use.
Yes, sir. Yes, sir. Thank
No,
you, sir.
no need to blot.
Okay, sir. Thank you. Thank you so much.
Sir, thank you. Thank you very much, sir. That's a wonderful presentation.
Thank you.
So, yeah, just a couple of, sir, is there like, is there any other, is there any other advanced technique like for detection? So we were speaking about LAMP assay, PCR and SSL, like say SSCP
Yeah.
and other things. Why not real time and other things like
That
we
is-
know that cost
that-
is one for sequencing, but why not real time?
That is also there, sir. But
Yeah.
of late in USA, people are using biolog identification. Biolog.
Okay.
There just you need to screen, you need to culture the bacteria and load 10 microliter or 20 microliter to ELISA plate and just scan that with an instrument.
Yeah. So
Instrument
like
give you
a
100%.
point of care.
Exactly.
Like a point of care, yeah.
Yes.
That I think that will be easy for people whenever they go for markers or when they wanted to take in lot.
But scientific details they are not aware of.
Okay. Sir, one more thing, sir. Like in SSCP, we were like I saw that you were using like we try to use like negative control water.
Yeah.
Whether there is a need for positive control.
It is always better now, sir.
Yeah.
To compare.
Yes. So we try to like when we in lab we try to put known like a mutated one
Organism.
variant.
Correct.
Yeah,
Correct.
yeah.
Correct. Anything can be used.
Yeah. Thank you, sir. Thank you
Thank
for
you.
the wonderful.
Thank you.
Presentation.
Thank you.
So, dear participants, anyone among you, please propose vote of thanks to resource person.
Hello.
Hello, thank you, sir. Thank you very much, sir.
Thank you,
Professor
sir.
Umesh, sir.
Yes, sir. Thank you.
Yeah.
Hello.
Yes, Niranjan Raj.
Sir, vote of thanks ಹೇಳೋಣ ಅಂತ.
Oh, ನೀವು ಇದೀರೇನ್ರಿ?
ಹೌದು ಸರ್
ನಿಮಗೂ ಬೇಕಿತ್ತ ಇದು?
Yeah.
Okay.
Sir, on behalf of this 15th Life Science Refresher course, I extend my heartfelt thanks to Professor Umesh for his very effective and informative speech. Although we have heard him many times, every time he comes up with new ideas, new concepts, and new things and refreshers about our research aspirations. Thank you very much, sir.
Thank you. Thank you.
Thank you, sir.
Thank you, Dr. Basarajappa, Dr. Niranjan Raj, and all others. Thank you very much.
Thank you, sir.
Thank you, sir. Thank you so much.
Thank you, sir.
Thank you, sir.
Yes, sir.
ಬಜೆಟ್ ಎಷ್ಟೇ ಕರೆಕ್ಟ್ ಗೊತ್ತಿಲ್ಲ ಲಿಸ್ಟ್ ಕೊಡ್ತೀನಿ ಕೆಮಿಕಲ್ ಲಿಸ್ಟ್ ಇದೆ ಬರ್ಕೊಂಡು
ಬಜೆಟ್ ನಾವು ವರ್ಷ ಇಷ್ಟು ಕೊಡ್ತಾರೆ ನಮ್ಗೆ ಜಾಸ್ತಿ ಕೊಡ್ತೀವಿ.
ಈಗ ನಾಳೆ ಮಧ್ಯಾಹ್ನ ರೂಪ ಇರಲ್ಲ ಅಂದ್ರು ಪ್ರಾಕ್ಟಿಕಲ್ ನಾನು ಮಾಡಬೇಕಿಲ್ಲ
ಬೇರೆ ಯಾರು
ಇಲ್ಲ ಪ್ರಾಕ್ಟಿಕಲ್ಸ್
Shall we leave, sir, or we'll have to wait?
Dr. Basarajappa.
Sir, please, sir.
No, participants are asking, shall we sign out?
Yeah, please. Okay, thank you. Tomorrow we can see you. Okay, thank you very much.
Thank you, sir. Thank you.
Thank you, sir.
Okay, bye. See you tomorrow.
Thank you, sir.
Thank you, Professor Umesh, sir.
Thank you, sir. Thank you.
Okay. Umesh sir,
Yes, sir.
ತುಂಬಾ ಚೆನ್ನಾಗಿತ್ತು. Ferocious. Okay.
Yeah. Bye.
Bye. See, thank you very much.
Yes, sir. Yes. Sir. Item number. ಯಾರು ಇದ್ದಾರೆ ಅಂತ ನೋಡ್ತಾ ಇದ್ದೀನಿ. Yes.